Good evening, everyone. It's good to be back once again. Welcome to our IVF webinar. It's wonderful to be back, and I'm glad to see some familiar names, at least right here. So that means that we are going strong with another week of events, and it's great to see you all here. Thank you so much for joining. And of course, as you can see, another guest is with us, and this is Roy Naja. Hi, Roy. How are you feeling tonight?
Hi, Caroline. Good.
It's great to
Nice
have
bank
you.
holiday and sunny bank holiday. So all good.
It's sounds great then, of course. Thank you so much for joining our My IVF Answers IVF webinars. As you know, there are plenty of webinars we have done so far, and today we are starting a topic on genetics. So this is definitely an interesting but also difficult subject. And tonight, of course, Roy is here to explain genetic health in female and male reproduction. So I'm really looking forward to see what you have prepared. And of course, we will start with Roy's presentation. And afterwards, as always, it will be time for your questions. So don't hesitate. Anything that's on your mind, just put those in the chat section, and Roy will definitely take care of those for you after the presentation. And let me just mention that Roy Naja, he's actually the lab director at IGENOMIX UK. So this is going to be for sure interesting, and I guess we can start off with our presentation. Okay, Roy?
Perfect.
Excellent. Let's go ahead then.
Great. Right. So I'm going to try my best in putting, the genetic and technical terms in layman terms and make it more patient-oriented, patient-friendly. So as Caroline said, I'm the lab director at IGENOMIX UK. We're a private genetic testing laboratory, a medical testing laboratory that specializes in pre-implantation genetics and fertility genetics. So let's carry on with our topic. Right. So genetic health and its what is the impact of genetic health on male sterility, female infertility, and the product of male and female fertilization, i.e. the embryo. So we're going to go briefly through these topics in this presentation, and we'll take some time and answer your questions at the end. Before it's important to give you a crash course on human genetics. So us humans were made of millions, actually trillions of cells, and in each cell we have one copy of what we call a genome, where we have all of our genetic code or material kept there. And this genome is divided into something we call chromosomes. We have many chromosomes in a human cell. And these chromosomes are further divided into little subunits called genes. And you might have heard that genes are these little genetic subunits that code for example, eye color, that genes in skin cells, they instruct the skin cells or the body to secrete this protective layer over your skin, the epidermis. Genes will instruct the body to form an organ, so on and so forth. So we are the product of genes. Now it's always ladies first, but I'm going to start my my topic in discussing the male factors of sterility or infertility, because there is a male contribution to infertility, and I've subdivided them into two major components. Defects that happen to the chromosomes, that are these big chunks of genetic material I just explained in the slide before, and defects that are that affect single genes, and both of these defects will contribute to male sterility or infertility. So a human a male, a normal human male will have 46 chromosomes or 22 pairs of non-sex chromosomes and a pair of sex chromosomes. And in the case of males, we have one X and one Y chromosome. So it's important to remember a karyotype of a human male is made of 46 individual chromosomes, or 23 pairs of chromosomes, where the sex chromosomes is X and Y. And chromosomal defects, which we will see in the next slide, what I want you to what's important from this slide is to so first of all, this is the WHO, this is the World Health Organization parameters for normal human semen parameters. So when you have a problem with infertility, you go to a clinic, they'll get a semen sample and they'll do a spermiogram or and where they analyze your semen factors. So I want you to remember that sperm concentration is very, very important in male sterility or male infertility. And you have to have 15 million of these sperms in one droplet, 1 ml. And if you have chromosomal defects, you're going to have a problem with your sperm. So 20% of all infertile or sterile men have sperm defects or semen defects. That have semen defects have chromosomal defects. So remember, those big chunks of genetic materials are chromosomes. And if you have a defects in your chromosome, you're going to have defects in your semen or in your sperm 20% of the times. And what you might hear when you go to a clinic and you have a low sperm count or not enough sperms in a droplet, you have something called oligozoospermia or oligospermia. And if you have no sperms in your ejaculate, you might have an ejaculate, but there's no sperms there. You have something called azoospermia or azoospermia. And this is known as male factor. So if you hear the clinician talking about male factor, the cause is a male factor to infertility or sterility, it's because you have either very, very low sperm or no sperm at all. And we'll see what causes that. So as I've said, chromosomal defects in your chromosomes will lead to a male factor, the absence of sperm or low sperm count. And there are two types of chromosomal defects. You might have a missing or an extra chromosome, which will lead to that. And Klinefelter syndrome is a very, very common disorder, not very common disorder. Most infertility or male sterility is attributed to Klinefelter syndrome in the category of chromosomal defects. So simply speaking, in Klinefelter syndrome, the male patient has 2 copies of the X chromosome instead of 1 copy, and 1 copy of the Y chromosome. So this will lead to infertility or sterility. Also, there is another syndrome where you have 2 copies of the Y chromosome. So if you go to a clinician, to a fertility clinic, to an IVF center, you've got a problem with your with infertility or fertility, they'll ask you to do something called a karyotype. A karyotype basically checks all your chromosomes. And if the karyotype shows that you have an extra X chromosome or an extra Y chromosome, this will explain the why you've got problems with fertility or why do you have infertility. Chromosomal defects also can be caused by structural events that happens in your chromosomes. Simply speaking, a male patient that has something called a translocation will have a low sperm count or no sperm at all. And this happens when a piece of one chromosome goes and sticks to another chromosome and vice versa. For example, here in between chromosomes 4 and 20, we've got a swap of genetic material. Now, the individual, the patient himself, is normal phenotypically, but when it comes to reproduction, there's problems. Either there's no production of sperm, or if you produce sperm, this sperm is defective. Another form of structural rearrangement is an inversion, where a piece of your chromosome gets just inverted. And this will show in a karyotype. And if you do a karyotype and you have something called a translocation or inversion, this will explain your infertility or sterility. Another common factor that leads to infertility in male patient is something called Y chromosome microdeletions. So also when you go to a fertility center or an IVF center, the clinician will ask for a Y chromosome microdeletion test besides the karyotype. And if you have these microdeletions, meaning small deletions in your Y chromosome, then you will have problems with fertility. You will either you will probably produce very, very low numbers of sperm. So this contributes to your infertility or sterility. Now we talked about defects in chromosomes, or these big chunks of DNA. Now we're going to talk about the defects in the genes, these little subunits on the chromosomes. So if these little subunits are altered in somehow, they will again lead to oligospermia or oligozoospermia and azoospermia, low sperm count, absence of sperm, or defects in how the sperm looks. So the sperm morphology and motility, how the sperm moves. This can happen. Defects can happen because of single gene disorders. And examples of single gene disorders are cystic fibrosis, sickle cell anemia, beta thalassemia, or myotonic dystrophy. I'm not going to go into details, but for example, I'll take the most common type of single gene disorder that causes infertility is cystic fibrosis. This is a disorder that affects multiple organs in your body, many organs in your body. And the main cause of death, it's fatal at a certain point. The patient might die because of suffocation, because you can't clear the mucus out of your lungs, and the patient has a respiratory arrest. However, if you've got this disorder, also you're infertile. So you have absence of the tube that takes the sperm from your from the testes to the outside of the body. The tube is called vas deferens. So this one is absent. So you won't have any sperms in your semen at all. And the other single gene disorders are sickle cell anemia and beta thalassemia. These are blood disorders. If you've got them, you have to do dialysis, so on and so forth. However, also your fertility or sterility is affected. So let's move to the genetic health in female infertility. So what are the genetic factors that contribute to female infertility? Again, they're the same, but we've got added causes that lead to the infertility. So we've got chromosomal defects, single gene defects, and the status of your uterus or endometrium, your womb. We'll go through each one of them. Again, like I've shown for the male, the female karyotype is made of 46 individual chromosomes and 2 X chromosomes, no Y. So if a female patient does a karyotype, we'll talk about the karyotype later, but I just want to show you here a quick overview of the female reproductive organ. And as you know, you've got your ovaries, where your follicles develop, or your eggs inside the follicles develop, and they're secreted in this tube called the fallopian tube, where it meets the sperm and gets, the fertilization happens, an embryo is created, and then it implants in the uterus or womb. So if you don't have any eggs produced, like you don't have any sperm produced, this is what affects female infertility. You have something called premature ovarian. You have something called ovarian failure. You might have oocytes produced early on in life. However, at a certain point, prematurely, you will cease to produce oocytes. So this is important in female infertility. So again, chromosomal defects could be numerical. You could be gaining or losing a chromosome, and the most common of those chromosomal defects is something we know as Turner syndrome. It's a female patient that has only 1 X chromosome, and the patient has deformities and very well known physical characteristics, but also infertility. Also, if a female patient has an extra X chromosome, instead of having 2 X chromosomes, it has an extra X chromosome, this also contributes to infertility. So this will show in what is called a karyotype. The clinician asks for a karyotype, and the karyotype might explain your infertility. If you have 1 X chromosome, 1 missing chromosome, 1 missing X chromosome, or 1 added X chromosome. Again, also, as for males, if you've got genetic material swapped in between your chromosomes, also you might have infertility. And in versions where chunks of the chromosomes are just inverted, you will get infertility. So these are very, very common causes of infertility, and you might hear of these translocation inversions or numerical abnormalities in your chromosomes, which causes your infertility. So again, single gene disorders also cause infertility, which are these little subunits on the chromosomes. If they are altered, they also will cause female infertility. And when we talk about female infertility is ovarian insufficiency or ovarian failure and premature cessation of the menstrual cycle, or amenorrhea. So single gene disorders that cause female infertility there's many of them, but the most important of them are listed here. Fragile X chromosome. This disorder is also is a disorder that affects, causes mental retardation or neurodevelopmental defects in certain females, but also causes infertility. Sickle cell anemia, another blood disorder that causes, you'll need dialysis in order to survive. However, also your fertility is reduced or is affected. Also, another common disorder is called galactosemia, where you cannot digest sugars in your food, and this sugar level goes up and then causes infertility. So these are just few examples of many single gene disorders or defects in single genes that can cause infertility. Now, also, the uterus or the endometrium is very important for female infertility, because you have this embryo that gets created in your fallopian tube, and this embryo needs to implant in your uterus. And the uterus has to be ready in order to accommodate this newly formed embryo. So this is another thing that might cause female infertility. And the uterus, as I said, needs to be ready to accommodate the embryo. So we talk about something called a receptive uterus or receptive endometrium, where it's ready to receive the embryo that is created. And that's very important. And also, your uterus or your endometrium or your womb needs to have something called good bacteria. It's a misconception when people think of bacteria, they think of infections. But in the human body, bacteria is very important. And two places where bacteria is very important is in your gut. You have to have your good bacteria in order to have good digestion. And for females, in the uterus or endometrium or womb, you have to have good bacteria for it to accommodate, to accept the embryo, to support the implantation, sticking of this embryo, and development of the embryo. So if you have failed implantation, sometimes it's because you have the nasty bugs in your endometrium. And there are tests out there that will check the receptivity of your endometrium, and if there's good bugs, good bacteria in your endometrium. And one of these companies that offer these tests, a very common test that we do offer, is at IGENOMIX, if there's something wrong with your endometrium. So also, now we're going to talk about the product of male and female fertilization. So a sperm meets the egg, or a sperm fertilizes the egg, and then an embryo is created. This embryo migrates into this tube that I showed you at the beginning, the fallopian tube, and goes and needs to be implanted in the uterus. Now, if an embryo, because once an embryo is created, this is a potential individual, and it needs all the number of all the chromosomes that a male or a female has. So if it's a male embryo, you need to have 46 pairs of chromosomes, where the sex chromosomes are XY. And for females, the embryo needs to have 46 chromosomes, and where the sex chromosomes are XX. However, this is due to biology. With advancing maternal age, some chromosomes are lost or gained in an embryo. So an embryo might be missing a chromosome, or might have extra chromosomes. And this definitely will lead to a failure of implantation or a miscarriage, and this is a main cause of infertility. So advanced maternal age is one of the most important reasons that lead to female infertility. And this goes with age, and I want you just to look at this blue curve, where you see that these abnormalities in the chromosomes of the embryo go up to 85% in females that are aged 44, 45. So this means that only 15% of your eggs are good. So if you have 10 embryos created at an IVF clinic, on the average, only 1.5 of them are going to be good if you're aged 44, 45. And this is statistics. And it's very important if you have an advanced maternal age, it's to seek help, because there is a solution. One thing that we can do is to screen those embryos that are cultured at the IVF clinics for missing or gained missing or gained chromosomes. And this is called pre-implantation genetic testing for aneuploidy, which was previously known as PGS. So what happens is that you go through an IVF cycle, you get your embryo created at the IVF clinic, then you do something called an embryo biopsy. You take a very small piece of the embryo. This will not damage the embryo if the embryologist is experienced. You freeze the embryo, you put this little piece in a tube, you send it to a genetics laboratory. The genetics laboratory will check if this embryo has extra chromosomes or missing chromosomes. And if yes, we will let the embryologist know that it's an abnormal embryo, do not transfer this embryo. And if the embryo is normal in terms of chromosomal complement, we'll tell the embryologist at the IVF center, yes, you can transfer this embryo. And this will help some patients tremendously, especially patients with advanced maternal age, achieve a normal pregnancy. And I guess that's it. And it's time for some questions.
Wonderful. Thank you so much, Roy, for that presentation. Lovely to find out a bit more on genetics. So thank you so much for explaining all the details. And now, yes, it is time for some of your questions. We don't have any questions yet, but someone is typing, so I'm sure you will have some questions. We just need to wait a bit longer for that. I would say it's more of a complicated topics. That is why sometimes we need to wait a bit more for those. So don't worry. Again, we just need to have a minute to see. And if I just may ask, so this is of course out of curiosity, how did you start working at IGENOMIX? If you can tell us in the meantime while we are waiting for those questions.
Well, I was working in fertility genetics for many years. I worked at the NHS. I worked at UCL for many years, and then I founded the lab in 2017 alongside Professor Alan Toner, which is a pioneer in the field. And IGENOMIX is a global company. We've got more than 23 different laboratories all over the world. It's one of the biggest of its kind in pre-implantation genetics. Yeah, this is I started with IGENOMIX in 2017 when we started.
All right, thank you for answering my additional question. Just was curious how it all started. It's good to know. And now we do have 2 questions for now. And of course, I do see people are typing in. So let's go ahead with those questions, okay? Let's have a look. First question from Margie. In which cases are genetic testing necessary for younger patients? Thank you so much for your question.
So if you have, so the cases where you have a repetitive implantation failure or miscarriage, then regardless of age, patients will undergo genetic testing, that the patient himself or herself. And the clinician will ask you for a karyotype. So this is the genetic testing. So irrespective of age, if you have repetitive implantation failure or miscarriage, you will get genetic testing. Margie.
Wonderful. Thank you so much for the clarification. Of course, the very first question. And let's have a look. Another question from Margie. Is it necessary when doing a donor cycle?
So if you're seeking donor eggs and you're at an advanced maternal age, the clinician might not ask for any genetic testing from yourself. Yeah, so depending on the age of the donor, if it's a young donor, the probability is that those eggs are going to be good. There's not going to be gains and losses of chromosomes, and you might not need genetic testing.
Excellent. Thank you so much for the clarification to this question as well. And question from Matilda, do you think pre-implantation testing will become the standard in the future for all cycles, regardless of infertility problems in the couple? Thank you for that interesting question.
It's very funny that Matilda asked this question because I was giving a lecture to UCL students. I still teach at UCL, and this was exactly the same topic we were discussing last Thursday. So at the moment, I don't think this is going to be applicable to, it's going to be common practice because the technique is very invasive and it's very costly. However, in the futures, there's a lot of research where you can where scientists are looking into making eggs from something we call the stem cells. So from your skin, if you take a piece of your skin, you can produce eggs in a laboratory, and then you can fertilize this egg in a laboratory. You don't need to stimulate the patient, give the patient a lot of hormones in order to produce a lot of eggs, and it's an invasive process. You go in with the catheter in order to collect these eggs. So all of this can be scrapped if you can create eggs from stem cells and then fertilize these eggs in a laboratory, and then you can just test these embryos. Why not? And you'll be able to test for many things in the future. So I see it happening in the future if this stem cell technology is perfected. Yeah, but at the moment, no, because this is a very invasive and costly procedure.
All right. Okay. Thank you so much once more for that explanation. And what are the costs for genetic testing?
Well, this you need to ask your IVF clinic. I can't
Of course.
tell you, but I can't answer this question.
Understandable.
The IVF clinic will charge you. We charge the IVF clinic, and the IVF clinic charges the patient.
Exactly. Of course. Thank you so much indeed. Okay, let's have a look. So genetic testing could be harmful for the embryo, correct?
No, genetic testing is not harmful for the embryo. What could sometimes be harmful for the embryo is the biopsy, taking the biopsy. And however, many IVF clinics have very good and experienced embryologists that have perfected biopsy, and biopsying an embryo at most times will not cause any harm to the embryo.
Wonderful. Thank you once again. Okay, next question from Sylvia. Can you explain more about the test used to analyze the status of the endometrium in case of failed implantation?
Yeah, that's very important. I mean, that's a good question. So this test usually looks at the genes expressed in your endometrium. So we look, for example, our test, which is the most common test available in the market. We look at 236 genes that have to be expressed in a certain way in order to make your endometrium receptive. So basically, how do you do this test? You do this test in a natural cycle. You go to the clinician. The clinician takes an embryo, takes a piece of not the embryo, sorry, takes a piece of this endometrium, an endometrium biopsy, and puts it in a tube, and we do the genetic testing. And this is done in your natural cycle at a certain stage in your luteal phase, or after your LH surge, or in a slightly medicated cycle. And then we'll test this endometrium, or this piece of the endometrium, and it's not a harmful process. You get local anesthesia, and you won't feel it. And we'll let you know if this endometrium is receptive. So if your window of receptivity is where it's supposed to be, which is at day 19, And we'll also tell you if your endometrium has the good bacteria to accommodate the embryo. So yeah, I hope I have answered your question.
Perfect. Thank you once again for another interesting question, and of course for your help with that. Let's have a look. More questions are coming in. So, if the ERA test was receptive in cycle, right? Can I perform a fresh transfer?
No, we don't do the ERA test in a medicated cycle. Yeah, so you have to replicate the same conditions where as the biopsy cycle. So if you do an ERA test, which is the endometrial receptivity analysis, in a natural cycle, you need to transfer the embryo afterwards, in a frozen cycle, at the same time that the ERA test showed that your endometrium is receptive. The clinician will explain all of that.
All right. Again, perfect. Thank you so much for yet another question. Let's have a look. Next question is from Mel. How long do I need to wait for the results?
A couple of weeks. A couple of weeks. 2 calendar weeks.
All right. Someone again is typing. We need a minute to check. Okay. Oh, okay. I think there is one more question right here. What are the alternative to embryo biopsy?
Okay, that's a good question. Now we've developed at IGENOMIX a test called non-invasive PGS or PGTA, where we don't biopsy your embryo. We just take some droplets where your embryo is growing, because the embryo is spitting out cells, and these cells will show 80% of the time if the embryo has gained chromosomes or lost chromosomes. So it's a non-invasive technique. We've just launched it last year, and it's an alternative option for invasive PGS, non-invasive PGS. Yeah.
That sounds promising for sure. Thank you so much for that question indeed. And let's have a look. Okay, one more question. How does PGTA and the other types of PGT test account for mosaicism?
So nowadays we use something called next generation sequencing. You're doing PGTA, and the platform that this NGS or next generation sequencing is very sensitive. It can pick up mosaicism, and we found out, this is internal data, that only 5% of the time an embryo is mosaic, meaning we're not sure if the embryo has an abnormality or not. And only 2.8% of the time this embryo will have a low levels mosaicism. So this embryo is given a chance to be transferred, but before transferring this embryo, the patient will undergo genetic counseling, where the genetic counselor will explain what are the risks of transferring a low mosaic embryo. But that's only 2.8% of the times.
Okay. Thank you once again for yet another question. And of course, let's have a look. Okay, there is a follow-up from the previous patient, okay? You mentioned that other technique option that might be available, right? So is it as good as PGS?
It's different. It's not a diagnostic. It's not a test. It's a test that gives priority to embryos. So if you have 4 embryos produced in an IVF cycle, and we do this non-invasive PGS, it will give you, it will prioritize the embryo. So embryo number 4 is the best, embryo number 2 is the second best. So it gives a chance for each and every embryo to be transferred. So that's its advantage over PGS, but it's only 80% sensitive as compared to PGS. So PGS will give a more precise answer to whether you have lost or gained a chromosome. So there's pros and cons for non-invasive.
Again, thank you so much, Margie, for your follow-ups as well. And for now, at least, it might be our final question. However, if you have any left, you know what to do. Just type those in the chat section, so Roy will be able to help you. And the question is from Mel again: does genetic testing create designer babies? What are your thoughts on that?
No, we're not. There's a difference. It's important to understand the difference between choosing what ended up from the parents into the embryo. So, for example, if mom has a genetic disorder, mom has 50% chance of passing this genetic disorder to the embryo. And when you do PGT or pre-implantation genetic testing, you screen for the good gene of mom, which is 50% of the time statistically is received in the embryo. So we're just looking what ended up in the embryo, and we're not changing anything in the genetic makeup. So if you've heard of gene editing, then it's actively changing your genetic material, and then you'll have designer babies. But with PGT, there's no designer babies.
All right. Again, thank you so much for that explanation. And Margie has added one more question. Does fibroid has to do with some female genetic issue?
There are certain studies that fibroids or I forgot the other word, the other scientific word for fibroids. Some patients with some gene defects are more prone to develop fibroids. But usually these fibroids are caused there's no genetic cause for these fibroids, and once they're removed, they should not cause to any infertility. Leiomyomas.
All right. Again, thank you so much indeed for yet another explanation. And at least at the moment, I don't see more questions. So, of course, if you have any, this will be like final call for them. But of course, as always, remember that if you would like to get in touch with Roy and his team at IGENOMIX, it's possible you will be able to use the link after we finish. You will be redirected to the site, and there will be an option for you to ask your questions. And if you wish to get more details, I'm sure Roy and his team will be happy to help you out. Someone is typing. Okay, Roy, so let's give it a minute. Let's see if we do have another question or it's a thank you. We will see. But before we finish, because we will be finishing slowly, so before we finish, is there anything at all you would like to add?
Me? No, there's nothing to add. So we've got another question. When are DNA fragmentation?
When are DNA fragmentation testing necessary? Mm-hmm.
So DNA fragmentation. So sometimes a very bad lifestyle, lots of smoking and drinking alcohol will lead to DNA to fragment in the sperm. So there are certain cases that the clinician will ask for a DNA fragmentation test to see if it's elevated as compared to a normal person. And there are some studies that associate a high DNA fragmentation with chromosomal abnormalities in embryos. So yeah, the clinician will ask you about your lifestyle and your diet, and sometimes they will ask for DNA fragmentation to be done.
Thank you so much for yet another question, Margie. Lovely questions and very on-point questions. So thank you so much for Margie, for you, Roy, as well. And I believe that was our final question, but there is also a comment I would like you to see. So wanted to say that I'm key KCL genetics student, and found this extremely interesting. Thank you. Thank you for that comment. We are happy that it was useful for
we
you.
Work with King's Fertility. The King's Fertility are one of our users.
Okay, good to know. Thank you so much.
To have educational events with King's at King's.
Great. Thank you so much indeed. And of course, we will be finishing for tonight. However, let me just again mention that Roy Naja was our presenter tonight. So thank you, Roy, for joining us tonight for your wonderful presentation. You brought lots of details, useful ones, and of course, for explaining all those answering and explaining all the questions that we've got from all of you. And as you know, it has been recorded. So if anyone missed anything, you will be able to watch this once again. It will be available on our website tomorrow, My IVF Answers. So you can go ahead and check it out tomorrow. And just let me also mention that we will be back here again. Tomorrow we will discuss PGTM. So join us at 7 p.m. UK time. And yes, that will be it for tonight. Thank you so much, Roy. Once again, it's been a pleasure to have you here. Thank you for agreeing to present for us, with us, of course. And all right, take care, everyone. And
Bye-bye.
I do hope to see you. Thank you, Roy.
Bye-bye.
Have a lovely evening, everyone.
You too.
Bye.
Bye-bye.